Welcome to 1.3, the cell, structure and function. What we are going to do today is we are going to go through the different types of cells from bacteria to eukaryotic. We're also going to look at cellular components and what their functions are. So we're going to look at the difference between plant cells and animal cells. So here we go. Different cell types and where they are found and what they do. But first, let's get into the hierarchy of life. Remember, we talked about the molecules, and these molecules, carbs, lipids, proteins, and nucleic acids combined to form our organelles. And so all those macromolecules we talked about will form the organelles that will function in the cell. So those organelles combine to form different types of cells. And those cells, different cell types, combine to form tissues. And various tissues will combine to form your organ systems, and then you get an organism. Okay, so cells is the basic unit of living things. You can have unicellular organisms, or you can have multicellular organisms like us, humans. So this is just to give you a relative size of different organisms, cells, and organelles to see where they fall. Most cells that we're going to talk about, the plant cells and animal cells, fall under the light microscope range at around 10 micrometers to about 100 micrometers. It depends upon the tissue that you're looking at. That will determine the size of that cell. Bacteria, on the other hand, they're relatively a little bit smaller at 1 micrometer. They're a little more difficult to see with a light microscope, but not impossible, especially if you have an oil immersion lens. An oil immersion lens can magnify up to 1,000 times. So let's look at the types of cells and where organisms fall in terms of domains. Now, this is the big overarching classification. Domain eukarya, of course, is everything that is a eukaryote. So this would be your plants, your animals. This would be your protists and fungi. Domain archaea is going to be your unicellular prokaryotes that live in extreme environments. They don't have any normal biochemistry. They may have some normal pathways, but most of these are known for doing some really unusual biochemistry pathways, metabolic pathways. And then bacteria, these are your unicellular prokaryotes that are your everyday bacteria. We find them everywhere. And these, when we talk about bacteria, these are the ones we're mostly talking about. So looking at the prokaryotic structure, the basic bacterial structure. Now keep in mind these are representative drawings. So not every bacteria will have pili, or not every bacteria will have flagellum, but some do. However, what they do have in common is that there are no membrane-bound organelles. What this means is there's no nuclear envelope, there's no endoplasmic reticulum, no mitochondria. It's just DNA, cytosol, nutrients, ions, amino acids, proteins, RNA, ribosomes. That's it. The metabolic functions will occur in the cytosol or in extended regions of the cell membrane. Their cell membrane actually will have folds in it to increase that surface area for those metabolic reactions. They do have a cell wall. Peptidoglycan is a carbohydrate with peptide cross bridges that it is made out of. So it is not made out of the same stuff that plant cell walls are made out of. Some bacteria will have a capsule. When we get to domains of life later on, we'll look more in detail at which bacteria have this capsule and which do not. But for right now, we're just looking at the general bacterial structure. Eukaryotes. Here is a representative drawing of your eukaryotic animal cell. So this is the generalized animal cell. Once again, remember most animal cells will have these organelles. However, there are going to be some cells that will have more of one type of organelle than another, and that has to do with that cell's specialized function. Remember, These are eukaryotes, so if it's a protist, it could be a unicellular protist, so it could be a unicellular organism, or we're looking at multicellular organisms in terms of plants and animals and fungi. These cells can be specific. They can be specialized for specific functions when parts of the tissue, like a glandular cell, would have a lot of smooth ER to do secretions or to secrete their products. Muscle cells will have lots of actin and myosin. That way it can have that contractile protein. Some animal cells may have cilia or flagella for movement. Cilia are short, tiny microtubule extensions off of the cell membrane. Flagella is going to be a long whip-like tail. And these are going to allow for movement. The centrosome area is interesting. This is the microtubule organizing center. The centrosome in animal cells has two centrioles. It is made up of microtubules. These centrioles, really not sure what they do. However, plant cells have a centrosome area, but plant cells do not have centrioles. So they can form the spindle, they can organize their microtubules, but plants do not have a visual representation of where this area is in the cell. So looking at plant cells, these are your organelles that you need to be familiar with. Once again, this is a representative drawing of a generalized plant cell. Some plant cells will have more chloroplasts than others. Think about your roots. Where are your roots? They are in the soil, so they don't have very many chloroplasts, if any. Okay, their job is water and nutrient acquisition, so they're going to have structures that are going to enable that function. Leaves, on the other hand, are going to be packed with chloroplasts. So some organelles that are not found in animal cells is a large centralized vacuole. This is going to be primarily filled with sap, and it's going to help maintain that turgor pressure on the cell wall. Another one is chloroplasts and plastids. These are going to store pigments and function as the site of photosynthesis if it's a chloroplast. And then the cell wall. Animal cells do not have a cell wall. Fungi, plants will have a cell wall, and some protists will have a cell wall. And this helps maintain the cell shape. Remember, big thing about plants is they can't get up and move around like animals. So they're more rigid, and that's one of it is due to their cell wall. So this is a generalized plant cell. Remember, we can have unicellular protists that have chloroplast or multicellular plants. So once again, plants can have specialized cells as part of their tissues. So let's look at a little bit more in detail on the nucleus. There are several components that make up the nuclear region. And inside, what we all know is that that's where the DNA is stored. Okay, it is stored in the form of chromatin. It contains the DNA and RNA protein that's involved. Chromatin, it contains DNA and protein that is involved in keeping the DNA folded and curled up and tied up. The nucleolus, this is the region that will make ribosome subunits. It is responsible for making the proteins in ribosomal RNA, and those substances can exit out of the nuclear pores. So the nuclear pores allow for substances to come into the nucleus and move out. The only thing that is not allowed to move out is the DNA. That stays in. But messenger RNA, transfer RNAs, proteins for DNA synthesis, those can enter and leave the nucleus through the nuclear pores. And it is connected to the rough ER. The endoplasmic reticulum, the red dots represent the ribosomes, but keep in mind there's going to be sections of the endoplasmic reticulum that do not have ribosomes. Okay. The nuclear envelope is also a double membrane. It is the only double membrane organelle in the animal cell. Both bacterial ribosomes and animal ribosomes are made up of two subunits. a prokaryotic and eukaryotic ribosome are slightly different in those subunits, but they are composed of ribosomal RNA and proteins. And they do function to produce the polypeptide chains by 4-protein synthesis or by protein synthesis. We'll get more into detail on that when you take your genetics course. And these guys may exist free in the cytosol or attached to the rough endoplasmic reticulum. But all protein synthesis will begin in the cytosol. If it's a protein for export, then that ribosome will have a chemical message that tells other molecules to attach the ribosome to the rough ER, but the protein synthesis actually starts in the cytosol, and then it's attached to the rough endoplasmic reticulum if needed. And that concludes our introductory into the cell structure and function. We are going to move now into the endomembrane system.